ago, this Belgian chemist revealed to the world a recipe for making mice. Scientists believed they could generate life as often as they wished.
If a dirty undergarment is squeezed into the mouth of a vessel containing wheat, within a few days, say 21, a ferment obtained from the garments and transformed by the smell of the grain encrusts the wheat itself with its own skin and turns it into mice which jump out fully formed.
Now science takes a different view. All life, mice included, evolved on Earth from a single primitive origin. What was the original recipe? And could it have occurred on other planets as well? In 1976, a spacecraft will approach Mars. It will make all the measurements we accept as usual in space exploration. But Viking's unique mission is to land on the Martian surface and look for life. It will be man's first attempt to find life on another planet. What will the lander be looking for? Trees? Elephants? Space monsters? What do today's scientists think about life outside the Earth? Carl Sagan of Cornell University explains.
Well, there was a time not so long ago when most scientists considered the possibility of life elsewhere as a kind of science fiction subject to be readily dismissed. You don't come upon such opinions so often anymore. I think the tide of feeling on the matter has changed somewhat, and people are willing to consider it as a significant scientific question. And there's a good reason for that. It's that for the first time we are able to actually explore the question. For thousands of years you could only guess. Well, now there are experiments that can be done.
The experiments Viking will do are designed not to find space monsters, but to analyze the Martian soil for microscopic life. Microbes cover the Earth's surface. The hope is that Mars is the same. In fact, the Martian experiments were designed entirely on our knowledge of the origin and development of the only life we know, life on Earth. We think the Earth and Mars are 4.5 billion years old. The history of life on Earth can be run backward, starting from the present day, 0 on the clock, towards 1 billion years ago. Geologists have attempted to track down the origin of life by tracing out the animal forms captured as fossils in older and older rocks. Before only 2 million years ago, man, as a species, is not found. Earlier than that, all other mammals disappear. Then reptiles go, fishes, plants, insects, shellfish, corals, and the earliest sea animals at just 600 million years ago. But earlier than 600 million years, less than 1/10 of the Earth's age, the geologists met a complete blank. There seemed no sign of life at all. Until, that is, this laboratory at Harvard began to take a closer look into the so-called Precambrian rocks, those older than 600 million years. By grinding down slices of rock until they were paper thin, Details never noticed before could be revealed. Under the microscope, they found obscure shapes, which might have been primitive single-cell organisms, but they were just shapes. There was no proof they had ever been alive. The search for proof led to this salt marsh on the Pacific coast of Mexico. The major inhabitants are blue-green algae, primitive single-cell plants. They'll grow in water virtually anywhere in the world, providing food for fish. But here, high salt content means no fish, so the algae accumulate into huge floating mats, disturbed only by a few birds. The algal mats are being studied by geologist Bill Schopf.
If you look in the fossil record, about 600 million years ago, you find advanced forms of life, and it had long been realized that there must be some evolutionary precursors. And this represented then one of the really classic puzzles in paleontology. Why was it that at 600 million years ago, one finds a vast array of complex organisms, but doesn't find any evidence of life earlier than that? Well, in the early 1900s, there were discovered, particularly by a man named Charles Walcott, a great American geologist, were discovered these laminated structures, rock structures that were made of layers and looked rather like large cabbages as much as anything. And Walcott suggested these might be formed by microorganisms, microscopic algae, for example. Well, there was a great debate about these things. This is about 1,000 million years in age, and as you can see, it's made of a set of layers within this rock. Well, these have been known for a long time. The question was, how were they formed? If I cut up one of these algal mats, you can see that it, too, is formed of very fine layers. And in fact, if we compare here the rock and the modern algal mat, they're very similar indeed. Well, it's been evidence like this, then, that has shown very clearly that the organisms, that there were organisms around in the Precambrian, these microscopic algae that form the modern mats, were the same organisms, we think, that formed the rocks.
With the link established between microorganisms and old rocks, fossils discovered in Australia, Canada, and South Africa have revealed signs of life going back over 3 billion years. Australia, 1 billion years old, highly developed clusters of cells. Canada, 2 billion years old, several different forms, most now unknown. South Africa, almost 3.5 billion years old, the oldest known fossils. A few shadowy shapes, almost certainly single-cell organisms, living in the same way as the Mexican blue-green algae. Biologists were amazed to learn that algae were flourishing over 3 billion years ago. These single cells use the same materials and life processes as all plants and animals, including man, alive today. The complete system must have evolved in 1 billion years. The cell structure consists of chains of protein, itself built from strings of amino acids, protein building blocks. But the amino acids need a construction blueprint, a genetic code hidden inside the cell. The DNA molecule, made of nucleic acid, carries the code, the basis of life. The nucleic acid code is made of millions of pairs of nucleotides arranged in a spiraling ladder. The order of the nucleotide rungs is the code itself. The code's function is to store all the information necessary for selecting and assembling amino acids into protein. These models, about 40 million times life-size, will show how a piece of the code is used to assemble a piece of protein. First, the blueprint splits. The nucleotides will only pair up in one way, red to green and blue to yellow. In this way, the latter is rebuilt in the same order. The code has been copied. Then the blueprint copy is joined by transfer units capable of reading the code, and each carrying a different amino acid. They try to find the point on the blueprint where they can match up correctly. If they fail, they are rejected. But if they fit, then protein building has started. Now the amino acids are making a particular protein, perhaps part of a blue-green algae. A different original code would have given a different amino acid order, a different protein. Three and a half billion years ago, this complicated system in use today had already evolved. Carl Sagan.
We find organisms as advanced as bacteria and blue-green algae. Those are quite sophisticated organisms, almost 3.5 billion years ago. Now, the origin of the Earth is only 4.5 billion years ago. And what's more, the early environment of the Earth was probably not suitable for the origin of life. We know that the moon, for the first 500 million years of its history, was terribly knocked about, probably by sweeping up debris from interplanetary space, and was melted all over its surface. It's very likely that the Earth suffered a similar fate. And it looks to me as if the Earth was not suitable for the origin of life until about 4 billion years ago. 500 million years later, you have very sophisticated organisms. So the origin of life had to happen very fast. I think something like a few 100 million years at the very outside, and probably a lot less than that.
The origin of life on the Earth is sandwiched between the oldest fossils and molten lava. Imagine the early Earth, soon after its formation from a spinning cloud of interstellar gas and dust, 4.5 billion years ago. Gradually, the surface cooled. Collisions with interplanetary debris became fewer. Hydrogen gas built up in the atmosphere, and water vapor condensed. Storm clouds began to gather. Somewhere on the surface, life started. Somewhere in a warm, drying rock pool, on a beach, or on the slopes of a volcano, the materials of life, amino acids and nucleic acids, were forming and beginning to group together. At the University of California, Stanley Miller is recreating the early Earth in a fourth-floor laboratory.
The primitive atmosphere was very different than the one we have at the present time on the Earth. The form of carbon was methane, which is natural gas, instead of the carbon dioxide that we have at present. The nitrogen was in the form of ammonia instead of molecular nitrogen as we have at present. There was water there just like we have now. But the major difference was that there was no oxygen and there was an abundance of molecular hydrogen. But nobody had ever tried an experiment to see what would happen in such an atmosphere.
20 years ago, when still a graduate student, Stanley Miller decided to do just that. It was to become a significant decision. He recreates the experiment now.
So the experiment was to construct a model ocean, which is the small flask here, a model atmosphere, which is the large flask containing the gases of the primitive atmosphere, and there was a condenser and a tube here which corresponded to the rain. And the whole thing consisted of a circulating apparatus.
So he boiled up the model ocean, added lightning, and set back to wait. After a week, the miniature ocean had turned a muddy brown and immediately became known as the primordial soup. To discover the soup's recipe, Miller set about analyzing it.
After I took the sample out of the electric discharge apparatus, I concentrated the material in that sample, then used a technique that by present standards is rather primitive, but was rather new at that time.
First, he placed a small drop of the mixture onto filter paper. The method then used solvents to spread out different chemicals in the soup. After spraying on a dye, he saw a collection of purple smudges. To Miller, the color and position meant only one thing. He had made amino acids, building blocks of protein.
The surprising part of this experiment, from my standpoint, was the large amount of amino acids obtained in the experiment. In fact, the major group of organic compounds produced by the spark were amino acids, and one had no right really to expect such a result.
It was a historic result. Anybody could make amino acids, but creating them this way, under primitive Earth conditions, suggested that scientists might be able to reconstruct the origins of life. At the Salk Institute in California, the possibility was recognized by another chemist, Leslie Orgel.
Stanley Miller had shown how you could make amino acids under conditions that could have existed on the primitive Earth, and so we knew how to make the pieces from which proteins are made up. We decided that we would try to do the same sort of thing for the nucleic acids.
Nucleic acid, carrying the genetic code, is as essential to life as the amino acids. Leslie Orgel's staff take their experiments out to the desert in search of conditions which could have existed on the primitive Earth. Hot, dry days alternating with cold, wet nights. Using only sunlight to heat simple chemicals, they've succeeded in producing the beginnings of nucleotides, nucleic acid building blocks. Then they try to combine the building blocks to produce just one side of a nucleic acid.
On the primitive Earth, we think these must have been many hundreds of units long. Our best efforts have produced pieces about as long as this. If each of the green and red pieces represents a nucleotide, we've succeeded in putting together pieces about 6 units long.
Pieces 6 long, under realistic conditions, is considered a significant achievement, but nowhere near the ultimate goal. DNA, 100 million units long in everything alive now. So at the University of Miami, the approach is totally different. DNA is too complicated, they say. Proteins are simpler. They've collected lava from the slopes of Mount Kilauea in Hawaii. In a laboratory, pure amino acids are dropped onto the hot stones. Under primitive Earth conditions, they're trying to assemble proteins directly from amino acids. The problems are explained by Sidney Fox.
The dilemma that has existed is the question of how did ordered sequences of amino acids come into existence in the absence of the very elaborate coding mechanism that exists today. The answer that we have found, and which has been verified in a number of other laboratories, is that by appropriate heating of amino acids, and this is done under geologically relevant conditions, the separate shapes, the individual shapes of the amino acids result in their joining in specific ways.
To assemble amino acids, Sidney Fox has worked out a sequence of events that could have occurred on the early Earth. First, hot lava meets primordial soup. Water boils off. The amino acids form a sticky brown glue. He calls it protenoid. The protenoid is washed off the lava, possibly by rain or the sea. This is the crucial step.
What occurs then is what had long been imagined as perhaps the most complex of processes in the series of primordial evolution. In a very dramatic way, the contact with water causes the protenoid molecules to assemble themselves.
Under the microscope, the assembled protenoid looked like this. Small transparent spheres about the size of living cells. But they weren't just shapes. Fox photographed a growth sequence running over 3 days. And here it looked like a cell dividing. Had this simple process created life? Compare the synthetic cells with this. This is a living cell dividing. The yellow mass in the center is nucleic acid preparing to split up, carrying its genetic code into both new cells. What's happening is that the DNA ladder itself splits, then uses its pairing up ability to make 2 identical copies of the code. Biologists call this replication. The synthetic cells haven't got the ability to transmit a detailed code. Although they could have existed on the primitive Earth, nobody can yet say how they might have developed. Nucleic acid's crucial role has made Leslie Orgel pursue the replication process.
We found that if we take a piece of nucleic acid, put it into solution, and add to that solution a set of the small units that have to be organized into a new nucleic acid, then these units will line up in the correct order on the template that we've added. Each red piece in the template picks up a green one, and each green piece picks up a red one. And so we organize reds and greens on the new structure in a way which is just complementary to the one that we start with. This, of course, is exactly what happens in DNA replication.
Is this alive? Is the matching accurate enough for a complete new code?
Perhaps only 1 in every 10 of the pieces that we have in the solution finish up in the correct long scheme. We have a very long way to go. We've got to make pieces much longer than this. We've got to be able to replicate them much more efficiently.
Orgel is after efficient replication because it's essential for the final subtlety of the living system, evolution, or improvement by mutation. This is a mutation. As the nucleotides match up, very occasionally a mistake is made. A mutation changes the code and so alters the organism itself. Here, orange matched orange, not blue. A major change would be fatal, but a slight change could be an improvement.
Well, the consequence of that improvement is you can start with a relatively simple organism and develop from it something much more specialized and much more complicated. We believe that the first organisms on the primitive Earth were not much more than molecules a few hundred units long, and everything that's alive nowadays has evolved from them because of this potentiality for improvement.
It's easy to make the molecules. We even have some hints as to the origin of the genetic code, but no one has started with a mixture of gases and oceans and wound up with some animal crawling out of the side of the test tube. That has not yet occurred and is unlikely to occur, because what's necessary for the production of something recognizably alive is evolution, and the secret of evolution is time. The origin of life happened fast, that at the very outside it took maybe a couple of 100 million years. 6 days was once a popular hypothesis. It's not excluded, but not more than a few 100 million years. By the way, if the origin of life happened fast on the Earth, then that suggests that it's a not uncommon event and that the origin of life could be expected on other planets as well.
This could have been a piece of another planet. It's a meteorite, formed at the same time as the solar system, out of the same materials. It's being examined at the Ames Research Center. Keith Kvenvolden.
The Murchison meteorite fell in Australia in September of 1969, and this rock was, or this specimen, the one you see here, was picked up almost immediately after the fall, and it gave those of us who are interested in organic compounds in meteorites an unprecedented opportunity to look for compounds in a situation where the specimen was uncontaminated by biology or by curators who, and for other meteorites, have put them in museum shelves and so on.
Under sterile conditions, they took the meteorite apart. Would it contain the materials necessary for life? Using sophisticated modern techniques, the sample was analyzed for a wide range of organic compounds.
We found a series of compounds, or a series of amino acids that was completely different from what had ever been found before by these previous investigators. And this again allowed us to say we had material that was different from the curator's fingerprints, but rather was material that looked like it had been produced during the meteorite formation process.
When I saw the results of the Murchison amino acid analysis, I immediately looked in the electric discharge mixture for the amino acids that they had found in the Murchison meteorite, and I found all of them in the electric discharge mixture.
This has far-ranging implications, because if this kind of process can go on here in this solar system, then it could have gone on in any forming solar system or any forming star system anywhere in the universe. And of course, for those of us who are interested in the occurrence or possible occurrence of extraterrestrial life, it means that life, at least according to the theory of chemical evolution, probably is not unique to the Earth.